Good morning. I would like to thank the organizers for the invitation to participate in this workshop. It's my pleasure to share a regulatory perspective on the safety monitoring in clinical trials evaluating finite duration hepatitis B therapies. Here is my disclaimer. I'll start with some general guidelines for non-clinical studies to support clinical trials, followed by safety monitoring and evaluation focusing on hepatic flares during treatment and upon discontinuation of therapy. I will then discuss some of the key benefit-risk considerations for risk assessment when a serious safety risk is anticipated. Starting with non-clinical safety studies, in general, non-clinical combination toxicology studies of an investigational new agent, when combined with an approved drug, are not needed unless preclinical or early clinical data for the new agent suggests a potential for serious synergistic toxicity when used with an approved drug. For clinical protocols assessing combination regimens with two or more investigational agents, we encourage early engagement with the agency to discuss whether combination toxicology studies are needed to support clinical trials for the intended combination regimen and to discuss the duration of those studies. International Conference on Harmonization, ICH, M3R2 guidance on non-clinical safety studies will be the generally applicable regulatory guidance. When developing safety monitoring plans for phase 2 and 3 trials, pre-specified safety monitoring plans should be based on non-clinical data, pharmacokinetic and pharmacodynamic characteristics, and early phase trial findings. Eligibility criteria for the trial should be reasonable to avoid risk to vulnerable subpopulations. For example, initial trials should focus on adult patient populations without cirrhosis. Evaluation of dose exposure response for safety and tolerability should be done to optimize benefit relative to safety risks and to inform dosing recommendations. Potential for toxicities with demonstrated or predicted drug-drug interactions should be considered in addition to potential for overlapping toxicities with the individual drugs in the combination regimen. Safety monitoring plans should consider mechanism of action of the drug, as there may be different safety concerns associated with specific drugs. Specific safety monitoring may be needed based on known safety issues in the drug class. For example, clinically significant immune-mediated adverse reactions have been reported with immunotherapies such as immune checkpoint inhibitors. Cytokine release syndrome represents serious adverse effects associated with T-cell engaging therapies. Severe thrombocytopenia has been observed with some antisense oligonucleotides. Any dose-limiting toxicities should be considered and adequately monitored. Dose modification guidelines should be clearly outlined in the protocol. Pre-specified stopping rules should be incorporated related to safety endpoints. Pre-specified interim safety assessments and ongoing review of data by independent data safety monitoring boards may help in making prompt decisions if new safety concern is identified. One of the unique challenges with hepatitis B trials is assessment of ALT flares. 3 different broad categories of flares were discussed at the FAASLD hepatitis B virus treatment endpoints workshop in 2019. To discern immune-associated flares from signal of potential drug-induced liver injury, serological and virological markers need to be assessed in addition to liver enzymes. As we all know, predicting the severity of hepatic flares is difficult. Close monitoring of ALT elevations is necessary to monitor trends over time. Criteria for defining and monitoring for hepatic flares During treatment and after stopping therapy should be pre-specified. Severe flares with deterioration in synthetic and excretory functions can be serious and life-threatening, particularly in patients with advanced liver disease. Adverse events of death or liver transplantation, hepatic decompensation, irreversible autoimmunity, or cases of severe hepatitis flare are major safety concerns. Now let's focus on systematic assessment of liver safety during trials. A working group convened by Hepatitis B Forum published consensus recommendations on evaluation and management of liver safety signals during clinical trials. I would like to emphasize that it is crucial that systematic data collection and full workup is done to rule out alternative causes of liver injury to enable a thorough causality assessment. Consideration should be given to convening an independent expert adjudication panel that may play an important role in assessing drug hepatotoxicity profile during clinical development. As we know, development of new hepatitis B therapies is targeted at finite duration treatment regimens. Severe acute exacerbations of hepatitis B may occur after discontinuation of therapy, particularly in the absence of surface antigen loss. Hence, treatment discontinuation should initially be evaluated in subpopulations with low risk of serious hepatic outcomes. Criteria for stopping therapy at the end of investigational treatment period should be well defined in the protocol and should be based on clinically validated biomarkers to ensure that the discontinuation of therapy does not pose undue safety risk to trial participants. Use of biomarkers such as HBV RNA and decline in surface antigen levels as a trigger for treatment interruption should be discussed with the FDA in advance of trial initiation. Patients should be monitored closely with both clinical and laboratory follow-up for at least several months after discontinuation of hepatitis B therapy. In certain circumstances, resumption of therapy may be warranted. Detailed plan or criteria for treatment reinitiation should be pre-specified in the clinical trial protocols. Scientific consensus is needed to formulate appropriate treatment in reinitiation criteria in the context of clinical trials. In general, off treatment when evaluating investigational finite duration therapies refers to discontinuation of all therapies, including background nuke regimens. There are serious concerns regarding stopping nuke therapy in virally suppressed patients on stable treatment regimen. To demonstrate sustained response off treatment, the duration of treatment consolidation period needed after surface antigen loss on treatment needs to be systematically assessed. This may vary based on the mechanism of action and half-life of a specific investigational drug. Criteria for stopping nuke therapy should be based on clinical evidence that also reflects current practice guidelines to avoid serious risks to trial participants. Long duration of follow-up is needed for patients who remain off therapy, both for safety assessment and durability of response. Known and potential risk of investigational drugs, as well as pharmacokinetic and pharmacodynamic characteristics, should inform additional safety monitoring and duration of follow-up. Now moving on to benefit risk assessment during FDA review process. In September of this year, FDA published a draft guidance on benefit risk assessment for new drug and biological products. This guidance articulates important considerations that factor into benefit risk assessments. Next 3 slides, I'll discuss some of the concepts outlined in this draft guidance. The benefit risk framework, as shown in this slide, provides a structured, qualitative approach for identifying, assessing, and communicating the important considerations that factor into the benefit risk assessment. The first two rows, analysis of condition and current treatment options, provide the therapeutic context. Benefits and risk management are product-specific roles for assessment informed by the evidence about the drug's safety and efficacy based on clinical trial data. Benefit risk assessment takes into consideration demonstrated therapeutic effect of the new drug and observed safety and tolerability profile in the context of the severity of the disease condition and the patient's current unmet needs. The benefit risk assessment becomes more challenging in cases where the potential for serious safety risk is identified or expected. For example, risks that are life-threatening or associated with significant morbidity. Some of the benefit risk considerations in cases where serious risks are anticipated are discussed here in the context of hepatitis B trials. Selection of a primary efficacy endpoint for which the relationship between an effect on the surrogate endpoint and the clinical outcome of interest is well understood to demonstrate meaningful benefit. For hepatitis B trials, HBV DNA suppression with or without surface antigen loss is considered a validated surrogate endpoint that has been demonstrated to predict clinical outcomes. Representing a specific important advantage over available therapies, for example, achieving surface antigen loss in a substantial proportion of patients. Potential serious risks or toxicities compared to safety and tolerability profile of available therapies. Example, safety profile of approved NUCs is well characterized. Demonstrating that adequate measures can be implemented to keep risk to an acceptable level in the post-market setting. For example, feasibility of monitoring in clinical practice settings. Understanding patient preferences and the potential acceptability of trade-offs between treatment benefits and risk outcomes is important to inform the benefit risk assessment. Moving on to risk assessment and mitigation measures. In regard to risk assessment, when planning the sample size and duration of a clinical trial, in addition to efficacy, consideration should also be given to the degree of precision that will be provided for evaluating an anticipated serious risk. Determining a subpopulation for whom the drug may have a more favorable benefit risk profile by identification of patients, example, utilizing a predictive biomarker who are more likely to experience greater expected benefits or less likely to experience serious adverse events of the drug. For example, consider heterogeneity of disease manifestations and progression in the patient population, such as patients in immune active phase versus immune tolerant phase. Prospective collection of data to evaluate a potential serious risk, such as by actively ascertaining the occurrence and nature of the adverse event of interest using targeted case report form, prompts, and/or independent adjudication. Implementation of appropriate risk mitigation measures into clinical trials with the ability to prevent or monitor for anticipated serious adverse events in order to provide sufficient evidence that the risk can be adequately managed post-approval in real-world settings. This slide highlights some of the resources available to the sponsors. Sponsors are encouraged to communicate with the FDA through the pre-IND consultation program. Sponsors can request formal meetings to get input for product-specific development programs. Various FDA guidances are available for reference as well. To conclude, during clinical trials, avoiding unreasonable or unnecessary risk to trial participants is paramount. Safety of a drug is assessed by determining whether its expected estimated benefits outweigh its potential risks in the context of the disease and available therapeutic options. Change in safety monitoring plan may be warranted based on emerging data. Hepatitis B is a disease of global public health importance. Collaborative discussions between academia, industry, regulatory agencies, and patient communities are crucial for efficient drug development and to mitigate risk. FDA remains committed to facilitate the development of safe and effective therapies for people living with chronic hepatitis B virus infection globally. And last but not least, from an overall public health perspective, FDA-approved vaccines are available for the prevention of hepatitis B virus infection. Thank you for your time and attention, and I'm happy to take questions during the panel discussion.